Hey everyone, my name is Isaiah, and for my final project for stem cell biology, I decided to provide an overview of the TGF-beta signaling pathway during palate development. Now, palate development is a complex process that involves two key components, the primary palate and the secondary palate. Now, with the secondary palate, we see that it develops on the left and the right side and develops vertically. But as you go through the course of palate development, we see the two palates, left and right, converge and form one singular palate. Now, when we talk about palate development, there are three key stages to note: palate shelf growth, elevation, and fusion. Here we see normal palate development in a mouse model. As we move from day 12.5 to 13.5 to 14.5, we see the movement of the secondary palate both on the left and right side move towards the center of the mouth. Now on day 15, we see a complete hard secondary palate that has undergone complete fusion. Now to dive into fusion a little more, the fusion stage of palate development is where the left and right secondary palate come together and touch. And then throughout time, they will fuse and create and become one full singular palate. Now, within the fusion stage of palate development, we have three steps: adhesion, which is where the epithelium of both left and right secondary palate come together and touch; epithelial mesenchymal transition, EMT for short, is where the epithelium dissolves, creating a mesenchymal space, uniform mesenchymal space. Now, keep in mind, this step is TGF-beta controlled. And then our last step of fusion is apoptosis and mesenchyme confluence. Now we're going to turn our attention to the inner workings of the TGF-beta signaling pathway during palate development. Starting with our ligands, we have TGF-3, TGF-1, and TGF-2. Aside from our ligands, we have our receptor proteins. These include TB receptor 2, which forms a receptor complex with TB receptor 1. And then we also have an accessory receptor protein called TB receptor 3. Now, when it comes to binding of ligands to receptor proteins, TGF2 has a low affinity for our TBR2 and TBR1 complex. Therefore, it requires an accessory receptor protein like TBR3 to help mediate that process so it can bind successfully to this complex. So that's what we see here. TGF2 comes in, binds to TBR3, it moves, and is successfully bound to TBR2, TBR1 complex. Now, if we're dealing with TGF1 or TGF3, those just normally come in, they have a high affinity for this complex, and they will bind successfully to this TBR2, TBR1 complex. Now, once the ligand binds, we see phosphorylation and activation of our TBR2 receptor protein. From there, the TBR1 receptor protein becomes phosphorylated by what we call TBR2 mediated phosphorylation. From there, a SMAD, either 2 or 3 signaling protein, will pass by, become phosphorylated, and therefore activated. Now, when this occurs, and once that SMAD2/3 protein dissociates with that phosphate group, we will see a dissociation of our ligands from our receptor proteins. Now, with the SMAD protein, the activated SMAD protein floating in the cell, its job is to now look for a SMAD4 signaling protein. So once it finds a SMAD4 protein, it will bind to it, creating a SMAD protein complex. Now, once it binds and creates this SMAD complex, the it will undergo nuclear translocation, which basically means that this complex will now be able to move through the nuclear membrane into the nucleus through a nuclear pore. From here, the SMAD protein will go out and search and bind to its specific transcription factor. And in this case, we're going to be talking about snail. Snail is a transcription factor that is vital for palate development. So once the SMAD complex detects snail, it will bind successfully and become activated. From there, it will target the specific gene and initiate transcription. Now, this transcription with this specific transcription factor will help the process that I've mentioned earlier, EMT or epithelium mesenchymal transition, where we see that epithelial tissue from the left and the right secondary palate dissolve and form a uniform mesenchymal cell space. Now, when a ligand binds in the TGF-beta signaling pathway, it also initiates SMAD-independent pathways as well, where the SMAD proteins are not needed. These include P38, ERK, J and K. Now, these SMAD-independent signaling pathways are initiated by MAP kinases. Now we'll turn our attention to cleft palate, a craniofacial development disorder where the palate fails to form completely. Here at the phenotypic level, we see various forms of cleft palate ranging from unilateral to bilateral. Now, if we look further at the cellular level with cleft palate, in general, cleft palate has been linked to mutations in 5 key components of the TGF-beta signaling pathway. These include the TBR1 receptor, the TBR2 receptor, the SMAD3 signaling protein, the TGF-beta 3 ligand, and the TGF-beta 2 ligand. Now, for my project, I decided to do some further research into a more severe phenotypic syndrome of cleft palate called Loy-Dietz syndrome, or LD syndrome for short. Now, LD syndrome is associated with heterozygous deletion mutations in the TBR1 and TBR2 receptor proteins. So what this means is this mutation causes an abnormality in the receptor proteins, which causes the TGF-beta 3 and TGF-beta 1 ligands not being able to bind. That's the end of the content for my project. Here is a figure key for my viewers to review. Here are all the figures that I've used throughout my video that you can reference with labels. Here are my references that I've got my content from. Here are my image sources. And that is all. Thank you, guys.